It's a great pleasure to introduce Casey Bryce from the University of Bristol. And well, continuing with an iron theme, but here looking at peatland iron cycling in a warming world. So Casey, over to you, please.
Yeah, thanks for the introduction and the invite, John. It looks like it's going to be a really nice couple of days. It's nice to see so many old friends in the chat as well. Hopefully we can catch up at some point. Yeah, I wanted to continue with the iron theme, but doing a bit of a summary of a lot of the work we've been doing related to iron cycling in peatlands, and specifically about why we think that might be important as the climate is warming and how that impacts a lot of aspects of biogeochemical cycling in peatlands in the Northern Hemisphere. So we're interested in peatlands because they store so much carbon. So Northern Hemisphere peatlands store about a third of all of Earth's soil carbon, and they're really common in regions where you have really waterlogged conditions, generally quite sort of cold or damp conditions in the Northern Hemisphere. And they're just able to build up these absolutely enormous carbon stores. The issue is that pretty much all of the peatlands globally are in a really, really sort of sad, sorry state. So that's historically been because of drying of peatlands for things like agriculture or mining or forestry. For example, even here in the UK, we still mine peat and export it for compost and sell it commercially here. But there's also a whole raft of new issues that are kind of superimposed on these historical ones, which are all related to climate change. So a lot of peatlands are based in permafrost regions, and we're seeing really, really dramatic permafrost thaw. For example, the Arctic's thawing twice as fast as other areas of the globe. And that's also leading to things like massive droughts and fires in peatlands. So we're seeing a lot of Arctic peatland fires, which is something that, you know, just shouldn't really happen in these cold, wet places. And the issue with that is that we're basically turning these peatlands from really important global carbon sinks into these really problematic sources of greenhouse gases and dissolved organic carbon. So they should be something that can kind of help with the whole climate crisis, but they're actually something that's causing a lot of problems. And this is a thing that people are really starting to realize. So there's a massive investment at the moment in trying to optimize peat restoration and also predict how peatlands will change in the future. And it was really encouraging that actually the last COP that just happened, that was the first time they had something called a Peatland Pavilion, which was essentially an entire area that was just dedicated to talking about peatland processes and how we can specifically answer these questions and work out how to make the most out of the kind of climate benefits that peatlands can provide. The issue with trying to optimize restoration and also predict how peatlands are going to change in the future is that they have absolutely massive biogeochemical variability. So there's just huge variation within them. So on one side of the scale, you've got sites like these. So this is a blanket bog in South Wales that I work on with Finn, who's one of my new PhD students in Bristol. And this is a really ombrotrophic system. It only receives water from rainfall. The nutrient concentrations, the iron concentrations, sulfur concentrations are really, really low, or often just completely below detection limit. So, you know, we have to try really, as someone who loves iron, we have to try really, really hard to detect any iron in a system like this. But then you've got peatlands on the other hand, like this one, which is a lowland bog that's actually on the Sandringham estate in Norfolk. And this is a minerotrophic peatland, so it has some input of waters that have an interaction with the surrounding rocks. And it so happens that this peatland is really heavily influenced by these iron-rich green sands that are really common in the Norfolk area. And then we have absolutely massive aqueous iron concentrations. So we're measuring aqueous iron 2 up to about 8 millimolar in these peatlands, which is, I don't know about anyone else who works in systems like this, but even for me who looks for iron everywhere, that is kind of mind-blowingly high iron concentrations. And if you pull these cores out the ground, you see that you've got, maybe I can put my pointer on. You've got loads of iron deposits, loads of iron minerals right the way through the core. The surface of the ground is just, you know, all our wellies are orange when we come out. And actually there's so much iron that when you have areas of bare peat, we actually end up forming these thick iron crusts that are at least 3-4 centimeters on the surface, and they're hard enough that you can just jump on them, which is something that's quite unusual if you're used to working in bogs. We also have a lot of sulfur cycling going on, so you can see you've got these, what we think are kind of elemental sulfur biofilms on these areas of bare peat. So that's just to say that, we're trying to solve this issue of how to best sort of manage and predict how peatlands will respond, but something that has an iron concentration of 10 micromolar is going to behave really, really differently to something like this, which has a massive input of things like iron and sulfur. And one of the reasons that we're really interested in iron cycling, specifically in peatlands, is because of how the iron cycle can link to the carbon cycle. And I would predict that this is not the last iron cycling diagram that's going to come up today based on the program, but I guess I get to do it first. So what happens there's lots of ways that iron cycling links to the carbon cycle. One of the main ways is that when we're oxidizing iron 2 to iron 3, we can form iron oxides and oxyhydroxides that can trap organic matter. So that could be via co-precipitation, you could have absorption of organic matter to mineral surfaces, or you could have the formation of iron organic matter aqueous complexes that would change the behavior and the bioavailability of that organic matter. But then when we switch from an oxidizing to a reducing regime, we're then able to reduce those minerals again, dissolving them, and also directly coupling the oxidation of organic carbon to CO2. So via iron reduction, we have a direct mechanism whereby we can produce greenhouse gases in a system that's rich in iron and carbon. And like I said, when we have this reduction, we also have a dissolution of minerals, which means that those minerals aren't there anymore to trap and stabilize the carbon that they were previously holding. So we can see a mobilization of carbon that was previously bound to iron minerals and potentially increase the bioavailability of carbon within a system. So as a geomicrobiologist, this is really fascinating to me because when it comes to the iron cycle, loads of the processes are microbially driven. So that's both on the oxidative side and on the oxidative side and on the reductive side. And they don't only influence the iron cycle directly, but via the different types of iron metabolisms, we can heavily influence things like methane emissions via, for example, iron-dependent methane oxidation, or something like N2O emissions via nitrate-dependent iron oxidation, which are all processes I could probably talk about all day individually, but will refrain from doing that at the moment. So when we look at lots of other systems, so lots of people are looking at iron-carbon associations in systems all over the world, and it's been particularly a big field in marine systems and looking at marine sediments. And we see that these iron-carbon associations are often responsible for trapping up to about 20% of total organic carbon in systems like that, in things like sediments, estuaries, forest soils. And one thing that we're really interested in is trying to understand whether these iron-carbon associations could also be important in a system like a peatland that just has so much carbon in it, where the fraction that we would have expected to be associated with iron might be a bit smaller. So one of the things we've been doing in relation to that is looking at iron-carbon associations in permafrost regions. And I want to show an example of some studies we've been doing on a chronosequence of thaw lakes in Barrow in Alaska. So what you have in a lot of permafrost regions is that when the permafrost thaws, you form these small lakes, and then over a kind of repetitive cycle, those lakes drain, and then soils and organic rich soils start to form on top of them. And what we have in Alaska is a chronosequence where you have some really young thaw lakes that drained pretty recently, they're maybe like 50 years old, and some that are really ancient. So we can have thaw lakes where soils have been developing for like 5,500 years. So one of the things we did, this was part of a master's thesis actually by a former student of mine in Tübingen, Hannah Joss, where we were interested in quantifying in a system like this, how much iron, how much organic carbon could we theoretically trap within these iron carbon associations. And the way that we quantify that, and the way that lots of people who work in lots of different systems quantify that, is by using a dithionite citrate bicarbonate extraction. So it's basically, it's measured via a kind of selective dissolution procedure. So you put the sample, it's pretty straightforward. You put the sample in a dithionate-based extraction buffer. The dithionite is a reductant, so you reduce all the iron minerals, and then that releases the carbon that was trapped by those minerals. Obviously, if you just put any kind of soil or something in a solution, you'll mobilize carbon that was never stuck to anything, that had nothing to do with iron. So we correct that by trying to work out if you put your sample in a solution that's the same pH and the same ionic strength, how much carbon do you mobilize? So we would subtract that from whatever value we get from this. And then we also need to correct that for the carbon that you have in the extraction buffer. So when doing that, we can make an estimate, and we think a pretty good estimate for how much organic carbon could be trapped by iron minerals in these systems. And Hannah got some really interesting results. So what we saw was not necessarily a strong predictor based on the age of the samples or based on the age of the thaw lakes, but we see that the amount of iron-associated carbon was heavily influenced by the soil horizons. So when you have a more mineral-based soil horizon, we have a bit less reactive iron and a bit less iron-associated carbon, whereas actually in the organic horizons, which we were a little bit surprised about, we have more iron-associated organic carbon and more reactive iron. And what Hannah's done is to color these little dots based on what is the percentage of the organic carbon, of the total organic carbon that's actually trapped by iron minerals in this system. And we see that on the whole, whilst it's quite variable for most of the samples, we're still coming out with a number of about 10 to 25%. So that's actually a lot of carbon in this very carbon-rich system that you can trap within these iron minerals. And we got a bit of a sneak preview of what these look like on the microscale. So we were able to get some nanosems images of these. And we can see that there's a really close spatial association between iron and carbon on the microscale. For example, you have these red iron oxides coating some particulate carbon, and you have some carbon that's dispersed within these oxyhydroxides. And we think that a lot of the reactive iron in this system is probably short-range ordered minerals like ferrihydrite. So what that tells us is that there's lots in these permafrost regions, you could have lots of carbon that's associated with iron minerals. But that doesn't really tell us about how we would expect this pool to change as the climate is warming. And it's not just us who've seen these these big iron organic carbon associations in permafrost regions. There's a few other groups also working in Barrow and especially working in Tibet who are seeing really similar things. So we think this is pretty widespread. So how's it going to change as the climate is warming? So we've been doing a lot of work related to that in a place called Stordalen Mire in Abisko in Sweden. So this is an actively degrading permafrost peatland that's formed on iron-rich lake sediments. And we chose it because it's within the northern circumpolar permafrost region that's really, really vulnerable. It's rapidly degrading, and we expect that there won't be any permafrost there by 2050. And this is all work of Monique Patzner, who just recently graduated, who worked with me when I was in Tübingen doing all this work in Abisko. So everything I present from pretty much from now on is all stuff that came out of Monique's PhD. So what the site in Abisko in Sweden looks like is something like this. And we've got these really defined stages of permafrost thaw. So we have these dry oxic palsas that have got ice underneath. They're raised up and the ice raises them above the ground surface. But as the permafrost thaws, what happens is that these palsas collapse and that you form this kind of bog-like area that is largely fed by rainfall and by runoff into the palsa, and there's still some permafrost underneath. What happens when the permafrost is completely gone is that we end up with this minerotrophic fen, where, because the permafrost isn't there anymore, there's a connection between the surface water and the groundwater. So there's actually a lake that's just off to the side here, which then starts to feed in and influence the fen. So it's a really nice model site because we've got all these different stages of thaw, but they're all within probably about 500 meters of each other. So we can look at different thaw stages really easily without, driving across hundreds of miles. So we expect by about 2050 at this site, we won't have any palsa anymore. And every year we go back, it's a sadder and sadder site. There's just less and less of it. So if we zoom in on what this gradient looks like, what we've been seeing is that if you collapse from these palsas through a bog and end up ultimately in a fen, you have an increase in the water content and a decrease in the oxygen. So we essentially have water logging during thaw. And Monique did some of these extractions similar to what Hannah did in Alaska, looking at how much iron-associated carbon do we have at different thaw stages. And we see that in these dry oxic palsas, if we look at the transitions between the mineral and the organic horizon, we have a lot of close association between iron and carbon. And this is an image, another NanoSims image of one of these associations from this sort of zone. And you see we have carbon in white and iron in red, and they're so closely associated, essentially completely kind of co-precipitated in this aggregate, that you come out with this kind of pinkish color. And again, when we see that as a percentage of the total organic carbon, it represents about 10 to 20% of carbon in those soil layers. But as we go through the thaw process, what we see is the amount of iron-associated carbon content rapidly decreases with thaw until we have essentially none left in the fen. There's no iron-associated carbon in the solid phase left in the fen. And we have a big increase in the amount of DOC that we have and the amount of aqueous iron 2 that we have. So the process that we see during thaw is essentially the thawing starts, you have water logging, you get iron reduction, and you're dissolving these minerals, which means that we expect these iron-carbon associations to be really highly unstable under future warming, and that potentially this thaw could represent an influx of extra carbon into the system, extra dissolved carbon into the system that could then be much more easily metabolized and released. So we wanted to know how quickly this iron-associated carbon is lost during thaw. So in that case, we're looking over the whole thaw gradient. So we zoomed in a bit in the next study that Monique did to just look at these collapse fronts. So this is just directly where the palsa is collapsing into the bog. And what we see is that the amount of iron-associated carbon we have is decreasing already before we even get to the bottom of these thaw features. So as soon as we initiate thaw, these iron-carbon associations are really vulnerable and we're just losing them straight away. And we find that at these thaw fronts, this is associated with a really big peak in DOC and in aqueous iron 2. So we see directly at the point where the pulsars have collapsed, we've just got a lot of iron and carbon coming out into solution. And if we look into the carbon, we see about 10% of that DOC is actually represented by acetate, which is highly bioavailable and potentially fuel for something like methanogenesis. We did a bunch of sequencing along this thaw gradient, and we did both extracted DNA and RNA and did some 16S. And we see that actually both the oxidative and reductive sides of the iron cycle are being enhanced by this thaw process. So even though we see a net iron reduction, we see an increase in both iron oxidizers, really classical iron oxidizers like gallinella and sideroxidins, as well as classical iron reducers like geobacter, increasing along this thaw gradient. And I'm maybe a bit old school when it comes to geomicrobiology, that I like to have things in culture before I believe that they do what we think they're doing. But we do actually have 2 iron reducing enrichments from this thaw gradient now. One is a geobacter and one's actually an iron reducing Cupriavidis species. So we know there's definitely a lot of microbial iron reduction going And we also had a little bit of a look at what this all means for greenhouse gases. And what we can see is that at these collapse fronts, we also have a good amount of methane being produced. Maybe good amount isn't the right phrase, but we have a big increase in the amount of methane that we're seeing being produced at these collapse fronts. And that actually correlates, if we look into the 16S data, with another increase in both methanogens and in methanotrophs. How much that's linked to iron cycling, we're not entirely sure yet, and it's something that is interesting and something we could definitely look into in the future. So the last thing I want to quickly talk about is what happens to these iron-associated carbon, post-thaw. So one thing we've been doing is to try and work out what the seasonal dynamics are in this system. And we did that with putting some redox probes out over the course of a whole summer. And what we can see is that whereas the palsa is always oxic, the fen is pretty much always anoxic. In the bog, we have a bit more variability. So it's a bit more susceptible to changes related to just rainfall conditions and temperature. And we actually did a little experiment where we put out some of these little Teflon bags filled with ferrihydrite. And we looked at how much the amount of iron in the bags changed throughout the season, so the early summer and the late summer. And we see that the palsas always accumulate iron, so they get more and more iron actually precipitated in them over the course of the summer. The fen always loses iron, so that's always reducing, as we would expect from the redox profiles. But interestingly, in the bog, we actually lose iron in the early part of the summer, and we regain it again in the late part of the summer. So that's showing us that there's a potential reformation of the loss of this trapped iron-associated carbon, depending on the seasonality within the bog. So to conclude on what we've been doing and all of that, what we can really see is that these iron oxyhydroxides are a potentially really important trap for carbon in peatlands and can influence carbon cycling. But we have hydrological shifts associated with thaw that stimulate a lot of iron reduction and then can release this iron-associated carbon. And in permafrost regions, this peak loss seems to be happening at collapse fronts. So it really makes the case for looking at transitional processes in thaw, not necessarily just kind of end members of thaw stages. But we see that there's a lot of seasonality and potentially important dynamics that happen post-thaw. We've started to think a bit about how widespread these processes are. So we've done pretty much all this work at Stordalen. And we've started looking at some other mires in the area. And we've actually been seeing, so this is with one of my new PhD students in Bristol, Anna Aberley. And we've been seeing that there's actually quite a big variation in aqueous iron content across mires in this area that are really close, like physically close by. And I think that's something that we definitely need to look into further and, try and work out how representative what we've done at Stordalen is over a much bigger area. So basically, I'll make a plug for, I think we need to know loads more about the mechanisms, dynamics, and the variability of iron cycling in peatlands if we really want to work out how to restore them and how to predict what they're doing. So with that, thanks to all the students who contributed to this, which was a lot, and loads of collaborators and all of our funders. And I'm not sure if I have any time for any questions. But
wonderful. Thank you, Casey. That's excellent. Really, really fascinating work and clearly globally important as well. There is a question that's just come in via the chat from BG. So it's a technical query. I'd like to know about the pore fluid extraction equipment which was used in the cover slide of the precipitate. I'm using rhizons for the same. The equipment showed you seem to be more efficient and can be used at the site itself, I guess. But is there any possibility to get the fluid from different depths using the same technique?
Yeah, that's actually a really good question. So that's something that Anna developed, kind of fueled by the fact that I'm kind of new in Bristol. We don't have tons of money. So, we try and like MacGyver a couple of things. But it's actually a really good system. So it's essentially a steel pole. That example is a steel pole, but we have them in plastic that we can actually acid wash and clean. that's got some slots at the end and a nylon mesh. And it is a lot faster than doing sampling with rhizons, but you have to filter immediately, which is then not as fast. So you can do a bit quicker pore water extraction, but then you have to manually filter. But if someone's interested in the design, I'm happy to talk to you about it, because we've been trying to get them to work for the last couple of weeks, and they seem to be quite good actually.
Thank you, Casey. Sorry, I missed it. There's a question from Mike Jeppen as well just before that. So, hi, Casey. Great work and presentation. Is the liberated acetate used for iron reduction or methanogenesis or denitrification? Any estimates as an N2O emission in addition to CO2 and methane?
Yeah, that's actually a really good. So we have been looking at that, and I didn't really have time to go into it in loads of detail, but it looks like the acetate's actually being used for iron reduction. So most of the methanogens that we see in the 16S are actually hydrogenotrophic methanogens, which is maybe not what you would expect when you've got so much acetate. So I think there's definitely some kind of competitive dynamics there with the methanogenesis and the iron reduction. We have been measuring N2O, but we didn't really measure much. But I think that there is more going on with the nitrogen cycle than we've managed to tease out so far. We've got quite a big peak in ammonium at those collapse fronts as well, and a bit of nitrate. We had a go at enriching some nitrate dependent iron oxidizers, not with a huge amount of success, but we also didn't try that hard to do it. So I think there's definitely something going on there that would warrant a bit more work.
Thank you, Casey. Thanks, Mike, for that question. Okay, so returning to the minerals, it's the JAVC. Have you looked into any precipitation of the Fe2 once it's released? So I guess siderite or something like that.
We haven't been seeing it. So most of the mineralogy that we've got comes from either we did a little bit of exafs and a bit of mossbower. And on these samples, neither of them were easy. They were both a bit messy, but it looks like we don't have something like, a lot of iron 2 minerals forming, but I think we probably do have quite a bit of absorbed iron 2. So that even you've got these oxyhydroxides and they are absorbing some iron 2, but we don't see any really distinctive iron 2 minerals where we think that there's this sort of iron carbon association. There is iron sulfides in the mineral soil, so the whole peatland is formed on these lake sediments, and they've got some FES in them. But we don't see a huge amount of that once you in the bit where we think there's a lot of important kind of iron carbon trapping. But there's more work we could do there. We've had a go. It's not been that easy.
Thanks, Casey. And one last question. This is from Ollie Moore. Do you have any idea as to the extent that methanotrophs could be sequestering some of the methane?
Not in terms of sort of rates or overall abundance. It does look like the methanotrophs aren't as abundant as the methanogens. So there's probably about 10% of the community at the collapse front is methanogens, whereas maybe only about 2% is methanotrophs. But I think we need to do some microcosms actually to quantify what are the rates.
Thanks, Casey. Great. Well, thanks everyone for the questions. Thanks for the excellent talk, Casey. Please, if anyone's got more questions for Casey, I'm sure you can post them in the chat, and Casey will try and get to them. And again, thanks for that really, really excellent talk.